Dear Chair and dear my colleagues, I would like to present my second presentation. The title of my presentation will be Biosynthesis of Silver Nanoparticles by using invasive Caulerpa cylindracea. Okay, now the outline of my presentation will be introduction, material methods, results, conclusion, and finally I will finish my presentation with the references part. Nanoparticles are of great importance in our daily life because we are using many different nanoparticles in many different sectors. The sample nanoparticles are silver, gold, palladium, iron, copper, lead, etc. These we can produce metal oxide nanoparticles and they are a very efficient material. However, when we use traditional method to produce nanoparticles, dangerous intermediates are formed. Therefore, we have to use green chemistry methods. We can use microorganisms, plants, algae, fungi, and biodegradable polymers to produce nanoparticles. In our study, we use Caulerpa cylindracea. As we might know, Caulerpa cylindracea is an invasive green marine seaweed, which is currently an invasive species in the Mediterranean Sea. In our study, we used the biomass of Caulerpa cylindracea to produce silver nanoparticles, and the experimental data was modeled by using artificial neural network. In Turkey, collection of invasive species is banned. Therefore, in order to carry out a scientific investigation, you have to take research permission from the Ministry of Agriculture. We did it, and then we collected our invasive species from Çeşme, Seferihisar, and Dikili, as you can see in the map. How we produce silver nanoparticles? First of all, after collection of Caulerpa cylindracea, we dried and we obtained powders by grinding the dried Caulerpa cylindracea biomass, and then we obtained the aquatic extract. Finally, when we mix the extract with the silver nitrate solution, we obtain brownish color in the solution. This will show us the formation of silver nanoparticles. And then we characterize the silver nanoparticles by using UV visible spectrophotometer, XRD, and FTIR. These are experimental conditions. What kind of parameters we studied? We changed the silver nitrate condition, Caulerpa cylindracea biomass, temperature, pH, agitation, dried, and time. So, of course, artificial neural network is of great importance in our daily life. Therefore, we wanted to use ANN to model our experimental data. The input parameters in our investigation was agitation time, rate, temperature, pH, and silver nitrate concentration, and the extract of the Caulerpa cylindracea concentration. On the other hand, we use absorbance value as the output data in our artificial neural network study. Okay, now I want to show you the results. When we look at the first figure, we see the effects of different extraction protocols. We obtained the best result at 30 minutes at 80 degrees Celsius. We also studied the effect of Caulerpa cylindracea extract concentration on the silver nanoparticle synthesis. As you can see from this slide, when we increase the extract concentration, we observed the gradual increase. And we studied the effect of time on the silver nanoparticle synthesis. Unfortunately, we observed fluctuation in this experiment, but this is well in line. Interestingly, this result is well in line with the literature. And we also studied the effect of different silver nitrate concentration on the silver nanoparticle synthesis by using Caulerpa cylindracea. As you can see, when we increase the concentration of the silver nitrate, we observe increased silver nanoparticles. These figures are related to effects of temperature and the pH. We obtained in more silver nanoparticle in basic area. I mean over than 0.7. And here you see the effect of agitation time on the synthesis of silver nanoparticles. You can obtain similar results when you apply 300 RPM. And also after synthesis of silver nanoparticles, we use some characterization methods such as UV visible spectra, as you can see here. And we also use FTIR spectrum. The results are well in line with the published reports in the scientific literature. We also use XRD to characterize the silver nanoparticles. The data are well in line with the literature. And we use the Scherer equation to measure the diameter or size of the silver nanoparticles synthesized. In this slide, you see the composition analysis of the silver nanoparticles produced by using Caulerpa cylindracea biomass. We observed the silver as a big amount because we synthesized the silver nanoparticles. And according to artificial neural network, we used the optimum hidden number as 17. The optimum percentages of training, validation, and testing was 85, 5, and 10 respectively. And we selected generally Levenberg-Marquardt backpropagation in our investigation. In order to check the performance of the artificial neural network, we drawn these graphics, predicted values versus experimental values. As you can see, we obtain high correlation coefficient. That means the developed artificial neural network works very well and define our data efficiently. As a conclusion, in order to prevent the harmful effects of the intermediates during the synthesis of nanoparticles, we have to use the green chemistry methods. They are actually very easy to apply. The use of toxic chemicals is limited and does not require the use of expensive tools. And we, during the synthesis of nanoparticles, we obtain lots of experimental data. Sometimes they are not linearized version. Therefore, in order to model these data, artificial neural network is an efficient way to model the experimental data. And finally, we can say that instead of eradication of invasive species, their biomasses can be used in the synthesis of valuable nanoparticles. These are the references related to my presentation. And finally, thank you very much for your attention.
Thank you, Dr. ÇAVAŞ.